Good evening, ladies and gentlemen. While we upload my presentation, I would like to thank the organizing committee of the conference. Most specifically, I'd like to thank Umberto Veronesi and Chiara Tonelli for being the soul of this project and have been so for 11 years. I'd like to thank most specifically Chiara, who organizing this conference, tried to tackle all the subjects involved with precision medicine with a truly interdisciplinary approach, something that would not usually be found because usually this has not happened, but as a matter of fact, precision medicine can only come to the fore and be successful at social and medical level only through a truly interdisciplinary approach. I shall be speaking about precision medicine, drawing from my two past experience. I'm a doctor and a life scientist and a scholar in bioethics. And I've also been dealing with the politics of science and sociology of biomedicine. Most specifically, I am expert on the involvement in people in medical innovation. So I'd like to contribute my two aspects of my experience on this subject so as to shed some light on the fact that we see here gather scholars coming from different walks of life to contribute to the success of this meeting. Now, I'd like to start going back in the past, as Michel Foucault would say, into the archaeology of medical science. We are lagging behind the schedule, so I'm not going to read this quotation, but it is important book on the birth of the clinic refers Foucault about the description of disease, and it was the middle of the 18th century. I think that you can read it by yourself. Here he refers to ghosts, shadows that apparently emerge from the body, and he compares this disease to another description that I won't again read, which is extremely specific. In this case, we are here at the beginning of the 18th century, and there is description of the brain and its structures, and you see that the different layers are clearly described with the colors, and I believe that looking at these two approaches to the body, I think that we understand what the space is, how we get to know our body, how we can distinguish what is normal to what is not normal or pathological, and who can tell us that a doctor in the 17th century could perceive this ghost? In 10 years, this ghost basically left, and a very clear vision prevailed. When did we see this clear aspect of things? When did we see things so clearly? When did it happen? Well, the space of diseased bodies, so this space of the configuration of the disease, is synonymous with the location of the disease on the body. So thinking about the disease as something only affecting organs and tissues is only a recent thing, because pathology again became very important only lately. So the 18th century doctor did not see what he saw, but that it needed several decades before the fantastic figures were dissipated to reveal in the space they vacated the shapes of things as they really are. This is the classifying medicine, as he describes it, where diseases are classified as different entities, distinguished entities, but the patient adds so many disturbances, his predispositions, his age, his way of life, and a whole series of events that, in relation to the essential nucleus, appear as accidents. This was a time when the disease was classified as such and an entity itself, but in its development in the patient basically was somewhat of a disturbance to the disease, because he would bring with himself all these accidents. Those describing a disease at the time had to describe all the signs that go with the disease, that characterize it, from the ones that are accidental, in that they depend on the temperament and age of the patient. So paradoxically, the patient is nothing but something exterior. So the medical reading must take him into account only to place him in parentheses. This is the quotation. So this is the world of classifying medicine that shapes much of the clinical studies that followed in those years. Let's have a look at this slide. That is the Precision Medicine Initiative as launched by President Obama. In this case, there's an individual, a woman in that case, that is clearly marked as distinct from the rest. And we already understood that genes, environment, and lifestyles are what characterize the patient, and in the past those would be considered as accidents, but now with precision medicine this is actually the focus of all the attention, and these factors become the focus of our attention, as you've heard by previous speakers, and as we're going to analyze during the next few days, whether there is any difference between precision medicine and tailor-made medicine. These actually are two different aspects. So what we should wonder is, to what extent is precision medicine to what extent is precise customized medicine, and to what percentage, to what extent is customized precision medicine? So Chiara was referring already to that. I believe that a certain gaze has now become possible, and that is the gaze which is connected to the digital aspects, the digital gaze. As you see here, there has been an enormous success of life sciences, as life sciences actually shape all the aspects of our time. And this is due to their technological essence, in that they describe all the levels of the organization of the living being in a digital way. So once this has been carried out, action can be undertaken on the living being using that digital circuitry that enabled us to understand the human being. Well, I believe that this is a very telling tweet that I was sent a few years ago, where basically typing 60 words per minute, 8 hours a day, it takes 50 years to type the human genome. So that's a 78 million of tweets. So basically we have the genome and the tweetoma, as it were. So the possibility to relate genome and the digital world, so much so as to speak about genome with tweets, because life has become text, because we've started getting to know life as text. As a matter of fact, the title of a book that I strongly recommend is Life Out of Sequence. So the life that emerges out of sequence, out of the sequencing of the DNA. So the study of life basically is understands life as a text. And this doesn't just happen with reference to genome. As you see here, there's a representation of what we might term as the cell to order, as it were. something that also Chiara referred to earlier on in her introductory remarks. So you see things that are available in many labs on the shelf, things that up until 15 years ago were quite unthinkable of. So there's a possibility to take any cell from our body and reconvert it into another type of cell using nothing but the digital alphabet that we've just learned. So these words basically correspond to nouns of genes regulating many other genes, and that in themselves enable us to shift a cell, to transfer a cell from being one into another. So much so that in specialized literature, specific reference is made to cell type plug and play. Plug and play cell, and this is why I've placed this image of an iPod, or that cells can be manipulated, reproduced as easily as we can perform a plug and play or develop an app, because we've already digitalized that biological function. So whenever you think about precision medicine, I should think that we have to introduce this concept, the concept of life scales. Scales obviously refer to the organization of different layers in the human being, so genomes, cells, epigenomes, organs, organisms, and ecosystems. All of them can be digitalized in that they can all be studied in a way which is relative to the DNA sequencing. You've already heard about epigenetics. So tomorrow you're going to hear more about that. So the study of epigenome can be a bit as the interface, reminding us of the environmental stresses that we've underwent, and transfer them into digital signs that can be then related to our genome. These are all a set of elements that correspond to the levels of our self. The genome, and it can be sequenced and then included in our Google server, and our cells can be reprogrammed and hosted in a biobank in New York, so that pieces of us, representation of our self, may interact with other players, with our institutions even. So I believe that the notion of scale is pretty important, because on the one hand, it helps us understand this huge depth when it comes to life sciences. Life sciences can explore all these different levels that are distinguished one from the other, but compatible, because they're all digital, or perhaps can be turned into something digital, better said. It also enables us to understand that to each of those levels, to each of those scales, what is important is the scale itself, i.e. the other level of the scale. So to what extent are we thinking big or small? And these are quite typical example. Precision medicine that goes across these scales is here represented as 1 to 100,000. 1 is the individual cell. One of the most important success of the past few years is our capability in understanding and analyzing genome, transcriptome, all those levels, basically at the level of one single cell. So you see that this is a liver, for example, and that is not important as such as an organ, as a liver, but it is scaled down to its individual cells, and each of those cells are important in themselves. And this is the opposite case. 100,000 British citizens are going to have their genome sequenced over the past few years. Interesting enough, this is a project of the National Health Service in the UK. And here there's another interesting aspect that tells us that these scales do not just refer to the life scales of the human being, but these scales interface also with those that we could refer to as governance scales, as political scales, i.e. these scales interact with being members of a political community. The UK, for example, states that they are sequencing 100,000 individuals so as to come about with better diagnosis and better treatment, and to carry out further research. What is innovative and useful about this concept of scale is that we see that as a parallel of these scale of power. This is Europe. And I'm showing you this because Europe is a continent which is getting reorganized. And as a matter of fact, it is within the European Parliament that they are deciding whether we shall have access or not to genetic tests. And as you're going to learn over the next few days, stem cell treatment is regulated differently if it is granted to individual states or if it is adopted at EU level. You may easily understand that this is not far-fetched. This is something that has a direct impact on the degree to which we can come to terms with precision medicine. The British project that I just referred to and the Obama project that is here represented in this image at the bottom refer again to the country. So you see that England is divided up into DNA bands, and this woman coming to the fore with her precise knowledge of herself is certainly an American woman because she comes forward with the backdrop of the US map. So they are biomedical scientific projects that are targeted to a national population. So these are truly political projects. Let's now move on to quote a couple of examples so as to make myself clearer. I shall be talking about cells and genomes. Now, to talk about genome, I'm showing you this drawing. This is an story commemorating the 10th anniversary after the sequencing of the genome, and I was strongly affected by this representation with each individual dot correspond to the genes, with that other representation, i.e. our guts, what lies in our tummy. And this sends us back to this representation. This is the anatomy class in the Padua University, where for the first time those guts were shown, to that space that we all have on our computer, because this is the browser that enables you to look at the human genome. Up until recently, the genome of some individuals was considered as the genome, but now you're going to have, all of you, your own genome. This is a book that I published with Erica Navotti, and basically we wanted to analyze the shift from the Padua University to the computer, where those guts were shown for the first time, to the time when on our monitor we can see each individual nucleotide making up our guts. So this gaze obviously has changed. And we also have to understand what is important and what is not. And we're going to see the implication of all that, looking at the important case that took place in Japan just a few days ago, seemingly for the benefit of the conference, and our possibility to interact with that. Many things change because in the meantime we shifted from a small place where the teacher would show the body to the happy few that were attending his class in Padua, to a case where many people, not everybody, but many people can have access to a type of knowledge which seems to be much more straightforward and immediate. And the decision maker changed, and the public, the groups of beneficiaries changed. This has changed. This is the cover of Time magazine that a few months ago contributed to, again, attaching even more importance to the concept of precision medicine. And this is very much the summary of all this. Here you have two women affected by brain cancer, And apparently we understand that the one is doing much better than the other. And why is one better than the other? Because the physician treating her detected that there's a gene mutation with one of the two patients, because for another disease, i.e. melanoma, a drug had been developed, a precise drug had been developed. So basically, the physician has administered that drug, and obviously, based on no evidence whatsoever, but the woman did better and is living longer. You have to understand that this woman is being treated not because she has brain cancer, or not because of what the pathologist said, but because we looked at her genes and we understood that probably it wasn't so much important that she was affected both by melanoma and brain cancer. The thing is that this woman basically has this gene mutation. So I understand that if I was successful first with this drug, I may be successful again with brain cancer, despite again the cancer not being treated with that molecule. So you may understand that the approach is completely different from the one that we saw looking at the quotations that I referred to at the beginning of my presentation. Let's now move on and look at cells. The most important change over the years has been the possibility to outsource the patient, as it were, i.e. to take a cell of a patient, reprogram it into different cells, EPS, i.e. Induced pluripotent stem cells. I stands for induced, and that refers also to iPod, iPad, iPhone, i.e. the I indeed stands for what is individual, and these pluripotent stem cells are such because they can be transformed into any tissue whatsoever. Now, on these tissues, many things can be made. For example, we can study the disease, and many believe that these cells can be used also to carry out studies. So rather than carrying out the studies on the individuals, we can carry out the studies on these avatars, on these stem cells that have been recreated artificially. Cellular reprogramming is certainly something that allows for this transition, and this is what lies at the very basis of the Obama initiative that has redefined his as a functional notation on the human genome. Because one thing is getting to know human genomes of 100,000 British subjects. But once you've known them, once you have understood whether those people are smokers or non-smokers, whether you've learned a whole set of data about these people, then if you cannot make experiments on that, if that specific genome cannot be tested with a whole set of experimental tools, well then you can do very little with all that bulk of knowledge. So what these technologies do is that they enable us to use such information and thus developing a bridge between the clinical data and the genetic lesions. And I'm going to refer to an example that is drawn from what we do in our lab, where two diseases are studies that have always fascinated us. So there's a small group of gene on this chromosome, and now we're not going to dwell on the details. There are some 20 genes that with some children are present with a pair less or with a pair more. So basically there's a problem connected to dosage on 20 out of 25,000 genes that make us basically so a pair is missing with certain children with a whole set of problems, among which mental retardation. But then they also have a very good speech talents. Indeed, these children were termed by the doctors as perfect cocktail party goers. The same 20 genes, if present with a pair less, are present with a pair more, well then other problems ensue, and that is autism. So you may very well understand that this is a pair of diseases where a change in the dosage of gene that is really minor can have important repercussions on human aspects that are completely essential for the human condition, i.e. language and social attitude. We so we made a group of these patients and we analyzed themselves and their family, trying to understand if taking a small cell of their skin, we could develop those pluripotent stem cells, the iPSCs. In vitro, we then developed a type of cells that were most relevant for their specific disease. In this representation, is the result of a student work. You can understand the scope of this research. So you may study one disease that is characterized by a certain set of features that cannot be studied because they lie within the body of the patient. So you just take a little piece of skin, you analyze the cell, you develop the induced pluripotent stem cells, and then you develop a model through differentiation. We wanted to show that there's a cell at the very beginning of its life, make us understand what are the molecular defects that are going to be amplified when those cells are turned into neurons or muscle cells or what have you. Obviously, the studies progressed. And on the left-hand side, you see the neurons that were developed in vitro coming out of the cell of the skin. And many labs, ours included, may then generate organoids, as we term them, i.e. an intermediate level between a cell and an organ in vitro, a mini brain, somebody termed them being, because these cells in vitro can assemble themselves, mimicking the brain. And this is very important also considering cancer. This tumoral organoid had an explosion in cancer studies because they paved the way to this possibility. That is, looking at a patient, and this is, for example, a case of a colon cancer, I can harvest a piece of tumor, for example, from this colon, and rather than studying these cells in themselves, because in the tumor, usually they're not by themselves, the tumor obviously affects tissue, the disease tissue, but a whole tissue. So I studied them not as individual cells, but as an organoid that in vitro tried to represent the same disease structure that existed in the colon of that patient. And out of that organoid, I can carry out a whole set of tests, and this is perhaps the most important for the patient. I can test in vitro on the artificial organoid of that patient the drugs that can be effective for that disease. So are we living at the time of avatars? We're basically taking something away from our body. We can actually outsource that so as to develop an experiment, a set of experiments to understand the interaction between genes and environment. Then there is another frontier, the one of Prometheus, in that we don't want to take these cells out just to study them or to test drugs on them. We want to do so to then reinsert them in the human body. And now I am approaching the end of my presentation. I'd like to refer to this case that took place in Japan and that has actually revolutionized precision medicine. I'm referring to Japan because Japan is the country that, again, was awarded as for the Nobel Prize of the reprogramming cells, and 1.5 years ago they started a clinical trial on degenerative maculopathy of a group of cells that are located on our retinas in our eyes. So that disease actually lended itself to be tested because, you know, the eye also lies somewhat out of the body. So it's an external organ, thus enabling us to monitor potential side effects. The idea was pretty straightforward. The idea was to take some piece of skin of these patients, reprogramming that skin into iPSCs, developing laminae of epithelial cells that the retina wouldn't producing any longer, reimplanting those in the eye of the patient. And that is the autologous treatment that doesn't require an immunosuppressant, so from my skin to my eye without any further disturbance. Nature Biotechnology has told us 6 days ago that when the second patient was being reimplanted, the study was interrupted. Why is that? Why was it interrupted? It was interrupted because sequencing the genome, so analyzing that deep body of information, 6 genetic mutations, 6 defects were identified in those EPCS. Well, we are not sure whether those defects were developed artificially or whether they're part of the original cell, because as a matter of fact we do have a genome, but that genome through our life changes, so each cell is slightly different from other cells. So to be on the safe side, when they realized that there was this problem, the trial was stopped, and the second patient was not administered that layer of on the retina. This is the magret eye, and as a matter of fact, this is just telling us that we can understand all in our genome, but that leads us to making very difficult choices. In Japan, for example, they decided to change the law so that they're not going to follow this model where basically I take away a piece of the skin, reprogramming cells and placing those cells back in the body. But now there's going to be a bank set up in Japan of cells taken by 100 or 200 Japanese citizens, chosen based on their genomes. Obviously, these people will all be administered some immunosuppressants. It will not be completely autologous, but here you see that there are straight correlations between the development of precision medicine on the one hand and the architecture of regulations. So the epistemology of politics, I would term it, i.e. who decides what. So much so that in Japan they have decided that these new treatments won't be marketed after the clinical trial, but only when we have tested there being 100% safe. As for the efficiency and effectiveness, it'll be the marketing that will see to understanding whether they are actually effective and efficient. And this obviously is completely different from the way that drugs experiments have been carried out so far in the rest of the world. So on the one hand, we learn that scientists think that it's going to be a routine, and not only to have the whole genome of all patients, but also to have such avatars interrogating them so as to put any question whatsoever. On the other hand, there's another colleague stating that these cells are still being developed as if they were Louis Vuitton bags, but the future is Walmart, so we have to mass produce them in a standardized way. So we don't have to develop something specific for each individual patient. We have to develop something standardized as they are going to do in Japan. So setting up banks of some 200 cells so as to solve the problem of all the local population. Now, four slides before I'm done. This is one of the first slides that Chiara showed us. And I'd like to quote T.S. Eliot. He says, where is the wisdom that we've lost in knowledge? Where is the knowledge that we have lost in information? So we always have to keep very alert when seeing this transformation. These have important social repercussions. Just think, for example, about how President Obama presented this initiative of precision medicine. He talked about another part of the American dream, the molecular American dream, where basically we can redesign our lot. And if it is true that we can redesign our lot, well, we may also say, as was anticipated by some of the opening speeches, the time has come, and this is my take-home message, to obtain an interaction between biomedical and social scales. The Obama initiative has been analyzed in terms of political studies so as to understand how to find how to find a result that is just and reproducible for all the citizens. I think that participation is important. We have to make sure that after decades where we've been saying that science has to be participatory, well, this initiative that touches each of us is not truly participated. This is an important study that was carried out in Los Angeles, who last year, this is an important empirical studies, when these two scholars referred to the 7 dimensions of participation. So whenever you're told that a certain project is participated, you have to ask those questions. What is the educative dividend? So how much people learned? What are the goals and tasks? To what extent people were involved in defining the objectives, controlling the resources, deciding to leave a research project if so they wanted, having a say in how the project was being carried out and in making this visible. The Obama initiative is a very interesting case. While we're speaking, it is still underway. As a matter of fact, discussions are still undergoing on the way precision medicine is going to be developed. And there are still some outstanding issues as to the Obama initiative. You see that for a certain degree, there's a high participation, but for other aspects, there's just a very cosmetic participation. Now, my take-home message again is the following. We have to think back to what we were saying earlier on, i.e. life out of sequence. Well, this is the example I quoted when I wanted again to refer to life with a digital gaze. And now I'm saying the same thing with other words. And now that we've understood how powerful digital is, we know that there are great potentials connected to this opportunity that we have. Now we have to understand what is included and what is excluded whenever we talk about precision medicine. So who is included, who is excluded? This afternoon we're going to talk about local biology because we've learned from anthrobiology that our bodies, lives have a whole set of specificities. So out of the sequence, we have to arrive at a result, the one that there's a Venice chapter. So to go back to one of the traditional achievement of this conference, i.e. a document that we hope to draft together with the other speakers and to all those who are interested, so that this conference, thanks to its interdisciplinary approach, which is pretty unique to this conference, trust me, so I believe that the document could be drafted that could catalyze speculations in a very tangible manner as to how to combine molecular and social studies. I'd like to thank all the foundations that enabled us to carry out our lab research and in STS. And I would like to thank again also the organizers, Chiara and Umberto, for making this possible. Thank you.